Hello, Professor and everyone. I'm Minsun, and today I'm going to present the paper on a T-cell resilience model associated with response to immunotherapy and multiple tumor types. First of all, I would like to introduce about the outline presentations. My content includes 5 parts, and at the very first, I would like to focus on background and rationales of the study. I would like to introduce to you some scientific terms relating to types of adaptive T-cell therapy that will be repeatedly mentioned during my presentation today. The first one was a tumor-infiltrating lymphocyte TIL. In this therapy, the T-cells are harvested directly from patient tumors, expanded in number in the laboratory, and infused back to the patients. And many of these T-cells naturally recognize and kill patients' cancer cells. The second therapy, called T-cell receptor CCR engineered T-cells, and the third one, CAR T-cells, are kind of similar. In both therapies, the T-cells are harvested from a patient's blood and genetically modified in the laboratory to have a new genes and encode proteins called TCR or CAR. And the T-cell also expanded in number and then infused back to the patient. Both are the modification target. T-cells specifically the patient cancer cells and trigger them to attack when they get there. To some extent, the cancer immunotherapies that act through T-cell have led to significant successes. Let's take a look to the immune checkpoint blockade therapy that block the CD8-4, PD-1, PD-L1 signaling. They can induce responses in cancer patients, but only 10 to 35% of patients within limited tumor types can benefit from this therapy, and they are called responder. In cell-based therapies, only 20 to 25% of patients with metastatic melanoma led to durable responses to tumor-infiltrating lymphocyte therapies, and the engineered T-cells only effective in hematological cancer but not the solid tumors. The same situations happen with other immunotherapies with very limited indications, limited tumor type, and limited number of responders. In fact, even after therapeutic interventions, the tumor microenvironment might suppress anti-tumor cells through different mechanisms. Let's take a look at the intrinsic mechanisms of resistance to immunotherapies of adaptive T-cells. The primary or adaptive or acquired resistance can be associated to alterations of signaling pathways such as MAPK, PA3K, WNT, and IFN, or could be the differentiations and with loss of tumor antigen expressions or alterations in antigen processing machineries or loss of HLA expressions or loss of target antigen expression, etc. And secondly, we may take a look at the tumor microenvironment, since there is always a balance between a large number of different immune cells, immune factors, and signaling molecules that determine the outcome of the anti-tumor immune response. They could be the good guys, which have rising the anti-tumor immunities, or the bad guys, which cause anti-tumor immunosuppressions, and listed here. So, under a microenvironment filled with various immunosuppressive factors, it's a very harsh battlefield for T-cells to fight against solid tumor. Most of them cannot persist under the pressure of such harsh environment. The term exhausted T-cells are used mainly to refer to effector T-cells with a reduced capacity to secrete cytokine and increased expression of inhibitory receptor. These cells were thought to be hypofunctional effector T cells that can differentiate from normal effector T cells and respond to a chronically high antigen load. So the question is there any T cell candidate that can still survive under such harsh environment and can be back into refreshed soldier that can continue to battle against malignant tumor? And the answer is yes. Several studies have identified and confirmed the existence of such T cells. They are characterized by highly cytotoxic state, proliferative activity, migrative capacity, and peripheral blood patients who respond to ICI therapy. This T cell can withstand the pressure of chronic tumor antigen stimulations and tumor microenvironment suppression without becoming exhausted, and importantly, they quickly expand with full cytotoxic functions upon ICI therapies. The author proposed using the term resilient T cells to describe these cells. And the word resilience in Latin means leaping back, which describes a state being able to withstand or recover quickly from difficult or stressful conditions. And this author also proposed the immune response in cancer therapies, and such are theoretical sphere of cancer immunology. As you can see here, the dynamic interactions of immune cell imprint and tumor cell role can determine the clinical outcomes of cancer disease and how it can be reshaped by cancer immunotherapy in terms of immune resilience. Therefore, developing new cancer immunotherapies are necessary. However, they require understanding molecular characteristics of T cells with high or low anti-tumor efficacies. In the past, several studies have collected single-cell T-cell transcriptome from tumor to identify the molecular signatures of T-cells associated with favorable or unfavorable clinical outcomes. An additional study on assessing the T-cell proliferations or cytokine level in a genome-wide script or upon reading from screen. However, they are not directly measure the anti-tumor capability, and in vivo genetic screen can address these limitations by pooling genes together to evaluate genes inhibition effect on the T-cell accumulations in murine tumor. However, murine tumor can have only a limited number of T-cell target RNA, and most genes will receive a zero count in a genome-wide screen and a draft from further evaluation, and may lead to missing many authentic regulatory T-cell anti-tumor functions. So to overcome the limitation discussed above, the study developed a computational framework named TRACE to identify the CD8-positive T cells that are resilient to immunosuppressive signal in solid tumor. This model is trained using the single-cell data from tumor, not subjected to any immunotherapy. There are two aims of the study. Is first, either to predict the clinical efficacy of immuno checkpoint inhibitor or similar therapies, and furthermore, to discover biomarkers and therapeutic target for T cells immunotherapy in solid tumor through the TRACE model. The TRACE model comprises of two different stages. The first one is to quantify the level of immunosuppression imposed on the CD8-positive T cells in single-cell transcriptomic datasets as the signaling activities of immunosuppressive cytokine. In particular, the first stage in the TRACE framework utilized the cytokine signaling activity that predicted by the cytoSIG database. The cytoSIG estimates the signaling activity of 51 cytokines based on the target genes motivated upon cytokine treatment collected from public transcriptomic data repository. Here we have the signaling activity of 2 to 8 immunosuppressive cytokines here and correlated to T cell proliferations. The first group, including the TGF-beta1, 12, and PGE2, show a significant negative correlation to T cell proliferations, and this is consistent with a rule reported in previous literature. The second group, including the interferon gamma and a tumor necrosis factor, do not show any significant negative correlations here. The third one, interleukin 10 and 4, although they show a significant negative correlations. However, previous literature report a very complex and inconsistent between the rules and the CD8 positive T cells proliferation. The final one, VEGF-A, even show positive correlations. So all of the later 5 immunosuppressive cytokines will be excluded from further analysis, which is focused on the first 3 immunosuppressive cytokines. In contrast to the T cell proliferations, the cytotoxicity score that's computed by granzyme and perforin expressions do not show any significant negative correlation with the cytosolic predicted signaling activity, and the expression level of the receptor do not correlate with T cell proliferations. So, in a later analysis, the TRACE model will focus on three key immunosuppressive signaling, including TGF-beta1, TREL, and PGE2, which modulate the T cell proliferations. As a further quality control, the author continued to evaluate the consistency between the TGF-beta1, TREL, and PGE2 signaling activities with each signaling node downstream activities to determine whether the cytosolic predictions is reliable or not. In case of the SMAD, which is a key mediator for TGF-beta signaling, and we can see the one with positive score of SMAD show greater score of TGF-beta1 activity in comparison with the one with negative score of SMAD. The same situations happen in the correlation between the PGE2 activity and CAMP. The author also showed the ROC curve for the quality of signaling activities in figure C and the quality of signaling activity across single cell cohort in figure D, and the correlation between the PGE2 signaling and CMP activity in figure F. They also suggest that the diverse cells population in tumor, such as the monocyte and cancer cells, should be the potential producers of these signals in figure G. And in conclusion, the cytosolic predictions is reliable, and the result could be applied in further analysis. The second stage of TRACE model utilizes a variable interaction test to identify genes expression of marker of T cells that are resilient to the previous identified inhibitive signals, including the TGF-beta1, TREL, or PGE2 and solid tumor. In other words, this model will identify which suppressive T cells are repressed or resilient under the activity of immunosuppressive cytokine. And the relationship between immunosuppression, T cell proliferations, and expression thesis of a third genes can be evaluated through the interactions covariate in linear regression. So I would like to talk a little bit about the regression analysis. Is it an important statistical method that allows us to examine the relationship between two or more variables in the datasets? Here we have one dependent variable, also known as the responder, the observations, or the Y variables, the main factor that we are trying to understand or predict. And we have several independent variables, also known as the predictor, the explanatory variables, or the X variable. They are the factors that we believe have an impact on the dependent variables. And to measure the relationship between these multidimensional variables, the multivariate regression is used instead of a simple linear regression. Multivariate regression is a technique used to measure the degree to which the various independent variables and various dependent variables are linearly related to each other, and it is commonly used as a supervised algorithm in machine learning, since it serves as a model to predict the behavior of dependent variables and multiple independent variables. And here's a formula that can explain how factors in variables respond simultaneously to changes in order and try to predict the output. To evaluate whether T cells with high expression of the gene Gs are resilient to immunosuppressive signal or not, the author utilized a variables interaction test in multivariate linear regressions. The suppression here indicates the immunosuppression level from TGF-beta1, TREL, or PGE2 signaling are predicted by using the cytosolics. Next, the proliferation score. This is computed as the t-value or test score equal the coefficient C divided by the standard error of the explanatory variables representing whether a T cells is proliferative or cytotoxic. Here the G represents the expression level of gene Gs across CD8 positive T cells in the tumor. And for each gene Gs, they perform the following regressions for each cytokine. The associations between the immunosuppression and proliferation is 8 plus C multiple Gs. They are typically negative because of high immunoexpression activities from TGF beta 1, TRAILs or PGE2 signaling result in a low T cell proliferation level. And with a positive gene expression level, a positive tau efficiency C will reduce the negative association between immunosuppression and T cell proliferation also, while the negative coefficient will enhance the negative associations. As a result, a positive TRAIL score for genes indicated that the T cell with positive expression for that genes maintain similar proliferative values even under suppression from TGF beta 1, TRAILs or PGE2 signaling, as the IL-7R examples here. And a negative TRAIL score for genes indicated that T cells with positive expression for the genes do not proliferate under suppressions from designaling, as the FREBP example here. Evaluate the TRAIL model. The author assembled 36 single-cell RNA sequencing datasets from more than 160 tumors, spanning 19 cancer types. Each tumor has a TRAIL signatures consisting of one TRAIL score for 8 genes. We can take a look at the figure 1D TRAIL score distribution across many cohorts. And the previously mentioned IL-7R and FIBP genes have consistently positive and negative TRAIL score across tumors respectively. Further, the TRAIL score for immune checkpoint genes are negative, while they are positive for the T cells persistent on activation marker here. In contrast, the TRAIL score for immunosuppression signal receptor are not significant. This suggests that the single-cell expression of this receptor may not be an indicator of cytokine activity. And finally, the TRAIL score from diverse cohorts are positively correlated with each other. So, using any one of 3 immunosuppressive signals, including TGF, beta 1, TRAIL, or PGE2, the TRAIL model achieves statistical significance with more than 0.1% genes with threshold for discovery rate FDR smaller than 0.05 in at least 130 out of 168 tumors. So the author will focus the analysis on these 130 tumors with significant TRAIL score. In the following part, the author will validate the TRAIL model in terms of both biological relevance and clinical relevance. They first validate the biological relevance of the TRAIL model by evaluating whether the TRAIL signature can predict the T cells persistent marker that was defined in previous study or not. As you can see, the TRAIL gene score successfully captured the top marker and the T cell persistent signatures shown here. They also found that the median TRAIL signatures merged from all immunosuppressive signals achieved the highest performance in comparison with other groups. The RC curve for the quality of the score remained around 0.8 and is statistically significant among different cancer cohorts. As a control, they applied the TRAIL model on transcriptomic data that's generated from in vivo cultures of cancer supply and found that the result performance is very close to 0.5 as a random expectation. To validate the clinical relevance of the TRAIL model, the author evaluated the TRAIL signatures on predicting the effectiveness of T cells in cancer immunotherapies. With the following procedures, to train the median trace signature using the immunotherapy knife single-cell RNA sequencing dataset that collected from public repository and use this median trace signatures in all following analysis. And in for the validation data, they use the T-cell back expression profile of the responder to T-cell therapy as listed here. When it comes to the clinical relevance validation result of the trace model, we can take a look at the trace signature correlations that predict the immunotherapy outcome in both pre-treatment and post-treatment tumor. Among the four candidates of signatures here, including the TRAS, the TSCM, which stands for the T memory stem cells, the T persistent, and the checkpoint. You can see the AUC of TRAS, the score, is generally the best among the four candidates. Although the T memory stem cells act better in 2 out of 6 cases. In this part, the author will evaluate the specificity of TRAS on modeling intratumor T-cell resilience to suppressive signals and counter-immunotherapies. As you can see, because the TRAS signature trainings use only the T-cell transcriptomes, so which are not enriched in tumor with low T-cell infiltrations. So, among tumor profile with high cytotoxic T-cell infiltrations, the correlation between the TRAS signature and back tumor expression profiles can reliably predict the survival outcomes. But in case the T-cell infiltration level here is low in back tumor, that the TRAS signatures become non-predictive. And similarly, when using the back tumor transcriptomes, the TRAS model that predict the immunocheckpoint inhibitor outcomes will be the highest accuracy when the intratumor T-cell infiltration is high. And as further controls, you can see the median TRAS signatures is not predictive of clinical outcomes when using T-cell transcriptomic profiles from peripheral blood immunocheckpoint inhibitor treated patients or tumor that treated with only chemotherapy. And also the TRAS signatures cannot predict the ICI outcomes using the box expression data from tumor with low expression of immunosuppressive signals. And in figure 4H, so you can see the risk score here is very high. However, far exceeding the expectations is that although the TRAS model was first developed to analyze the solid tumor, the median TRAS signatures is predictive of anti-CD19 CAR T response in hematopoietic malignancy. As you can see, the correlation between the TRAS signatures and the T-cell transcriptomic profile from infusion products in figure 2A, B, or before the CRT in figure 2E is significantly predictive of therapy response. So in nutshells, when compared to other published signature of T-cell anti-tumor efficacy as shown in figure 2C and extended data figure 4D, the performance of the median TRAS signatures is consistently the best at predicting the binary response. Using the ICI pre-treatment tumor sample and CRT infusion product, or survival durations using the pre-manufactured sample of CRT or TLI therapies shown in figure 2F. And the results suggest that TRAS could be used to screen a patient before the start of immunotherapy. It is very meaningful to applications because the immunotherapy is very expensive and is much more better if we can predict if patients will be benefit from immunotherapy or not before deciding to start an immunotherapy. Finally, make some comparisons between the results and results from other study. As you can see, currently the most well-established biomarker for immune checkpoint inhibitor are tumor mutation burden TMB, and tumor PD-L1 level assessed by immunohistochemistry axes. Regarding result of the first study, the AUC of both biomarker is recognized around 0.65 and 0.69 respectively. And in the second study, the AUC of PD-L1 TMBs was around 0.63. However, the mean of AUC across 6 datasets of this study is much more higher, around 0.855. However, such comparison is limited by a small number of T-cell transcriptomic datasets and indirect comparison across different studies. The next step, being encouraged by the performance of tests in predicting the T-cells' effectiveness, the author continued to identify candidate genes, regulatory, and therapeutic targets from test signatures. There are prioritized genes whose expression levels are consistently associated with worse T-cell tumor resilience and poor immunotherapy response in solid tumor. So the analysis only focused on a negative marker of T-cell effectiveness. And among all human genes, the FIBP ranked first as a negative marker of T-cell effectiveness. The test score of FIBP also consistently negative across single-cell datasets. As you can see here in figure 3a and 3b, the FIBP expression level are significantly lower in T-cells from anti-PD-1 responder than value from non-responder in solid tumor. And in figure 3c, among tumor sample obtained before expanding the TIL for data transfer, a high FIBP expression level indicates a worse in overall survival outcomes after adoptive TIL therapy. And in figure 3d, a high FIBP expression in both tumor transcriptomic data correlate with T-cell suspensions. The rest part of this study will focus on the wet laboratory validations of FIBP genes. The first two parts that will focus in the anti-tumor efficacy of FIBP knockout T-cells, both in vitro and in vivo experiment. The third part, the author suggests that the FIBP knockout limits cholesterol metabolism in T-cells, and in the final part, they will validate the inhibitions of T-cell functions by cholesterol rely on FIBP. So, to identify whether knocking out top 10 market genes identified in figure 8 using the script systems could enhance the anti-tumor efficacy of T-cells or not, the author cultured the mouse T-cells that were engineered with PMEL1, and PMEL1 are engineered T-cells that can recognize the GP100 antigens expressed by the B16 MIGP100 cell line of mouse cell line. After that, the cancer cell growth will be measured by the incucyte, and the cytokine release will be measured by ELISA. We can take a look at the knockout result of the third marker genes. Each gene was knocked out through a mixture of 3 independent guided RNA in the PMEL1 mouse T cells, and the abundance of the P16 MHGP100 co-culture with gene knockout T cells was measured at the intensity TD tomato. The y-axis present the difference between each target gene knockout and ROSA26 at the knockout control. As you can see, among this top marker, only FIBP knockout consistently promoted T-cells mediated cancer killing compared to other controls. Similar to the mouse model, in the human system, the engineered T-cells, which are positive for the NY-ESO1 antigens, was co-culture, with 2 kinds of human melanoma cells were also positive for the same antigens. Next, the author will perform the Western plots to prove the efficacy of Krebs knockout for FIBP genes in both humans and mouse models. Since previous genomes were screened on T-cell proliferations and interleukin-2 productions, revealed the CBLB as the most significant hit. So in this model, the CBLB will serve as a positive control, and the beta-actin will be served as the loading controls. The author utilized intusite imaging systems to evaluate the cancer killing efficacy of T cells that co-culture with the tumor cells that are labeled with the TD tomato and FBIBP knockout with 3 dependent guidance RNA 1, 2, 3 all led in decrease in number of cancer cells in comparison to the negative controls AAVS1. So, further analysis of the FIBP knockouts DE positive T cells confirmed that in both human model, as shown in figure 4D, and mouse model as shown in figure 4E, the T cell with FIBP knockout kill cancer cells at a higher efficacy than the control knockout. And moreover, in both human models on the left side and mice model on the right side, as you can see here, the FIBP knockout also enhanced the release of T-cell effector cytokines such as the interferon gamma and the tumor necrosis factors. In comparison to the AAVS1, which served as a negative control, and CBLB that served as a positive control, as you can see, the concentrations of FIBP is very close to the positive control CBLB. In the next part, they will use mice model to discover the anti-tumor efficacies of FIBP knockout T cells in vivo. Firstly, all the mice were subcutaneously injected by the B16M8GP100 melanoma cells. After 10 days of tumor implantations, the mice were sublaterally irradiated to get a lymphodepletion and were randomized aside into 5 different treatment groups, include the FIBP groups, knockout, the ROSA26 knockout a negative control, the CBLB knockout as a positive controls, and 2 different groups as wild-type T cells, and no treatment control that will be treated by saline injections. Running concurrently with this experiment, the T cells will be treated by the C8S9 INRP during 7 days, and in the next day, this mixture will be intravenously transferred to the mice. And after that, the recombinant human IL-2 was given by intraperitoneal injection twice a day for 3 consecutive days. After 2 weeks after the T cell transfer, the tumor site was measured in a double-blind manner twice a week, and the tumor area was calculated as length multiplied by weight. So, as you can see, there is no significant difference in terms of tumor area between the different treatment groups recognized at day 0. However, after 40 days post-HCT, and compared to no treatment control, the wild-type T-cell adaptive transfer significantly reflects the growth of GP100 positive tumor, and the ROSA26 knockout cell did not have lower anti-tumor efficacy than parental T cells, as you can see in figures 7C and 7D. In figure 5B, C, Ds, as you can see, In comparison to the ROSA26 knockout T cells as negative control groups, the FIB knockout T cells significantly reduced tumor size, and the overall tumor size reduction effect of this group was comparable to the positive control CBLB knockout T cells before any mice reaching endpoint, and both the FIBP and CBLP knockout T cell recipients have significantly longer survival than the ROSA knockout T cell recipients. So these results suggest that FIBP knockout is a potential therapeutic approach to enhance the adaptive T cell therapy. So to get deeper mechanistic insight into FIBP inhibitory effect on T cells, continue to perform the RNA sequencing in murine for the two groups. The left-hand side were the ROSA26 knockout T cells, and the right-hand side is where the PML-E1 FIBP knockout. Next, the IPA analysis revealed that all type enrichment on FIBP knockout are related to the downregulation of cholesterol metabolisms. And let's take a look at figure 6C. In comparison to ROSA26 knockout groups, the FIBP knockout groups had downregulated expressions of multiple cholesterol biosynthesis enzymes, such as the XMGCS1 or the SQLE or the cell-triggered receptor LDLR. However, the genes responsible for the cholesterol expressed from ABCA1 was upregulated in the FIBP knockout groups. So to further investigate association between the FIBP and cholesterol metabolisms, they also analyzed the correlation between the FIBP and the cholesterol pathway genes across the glycemia critical sample from the mice projects. Here, the results was used shown in extended data figure 8A and 8B. As you can see, the FIBP expression is positively correlated with positive regulatory cholesterol metabolisms and negatively correlated with the negative regulator. And further, when using the public chromatin immunodistribution chip sequencing data to identify the target genes of SREBF2 as known as a master transcription factor for promoting the cholesterol metabolisms. They also found that SERBF2 as the target genes to be significantly down-regulated upon the FIBP knockout. Next, the altered expression of several identified cholesterol pathway genes, including the ABCA1, LDLR, and SERBF2, was evaluated using the qPCR. As you can see in both mouse and human model, in the ROSA26 and AAVS1 acting as negative control, there is no difference between the expression level of these genes. However, in the FIBP knockout groups, there are significant decrease in expression level of SREBF2 and LDLR, while there is a significant upregulation in expressions of ABCA1. Next, they will use a Western blot to confirm the qPCR results. And as you can see here in figure 6E, there is a decrease in expression of LDLR proteins and SREBF2 proteins, both in the activated full length and the activated form. Conversely, in figure 6F, the FIBP overexpression T cells led to increase in expression of cholesterol metabolism regulatory at both mRNA and protein levels. as you can see in figure E. So based on the above result, we can come to a conclusion that the FIBP overexpression in T cells led to increased expression of cholesterol metabolism regulator at both mRNA and protein levels. And consistent with this result, the in vivo and in vitro cholesterol level in FIBP knockout T cells was significantly lower than level in the rows of 26 knockout T cells and negative control groups. As also here, the cholesterol level in T cell increased significantly up on the FIBP overexpressions shown in figure 6H. However, we do not observe any cholesterol level change in CBLB knockout T cells in figure 6G. Previous study demonstrated that the CBLB inhibition enhanced T cells anti-tumor efficacy by promoting the interleukin-2 productions and autocrine signaling. However, the FIBP knockout does not alter any interleukin-2 production in T cells. So, this data suggests that inhibiting different genes target may promote T cells anti-tumor efficacy through orthogonal mechanisms. So in the final part, the author will design some experiment to prove that the inhibition of T cell functions by cholesterol relies on the FIBP. First of all, the cancer-killing efficacies were determined during the co-cultures assay. As you can see in figure 6I, in figure 6I left, the cholesterol treatment of T cells significantly inhibited cancer-killing efficacy in ROSA 26 knockout groups. However, in FIBP knockout T cells that retain the cancer-killing efficacies even after cholesterol treatment. The data are consistent to the previously study. And furthermore, in the extended data figure 9E, as you can see, the FIBP knockout resulted in a marked reduction of LDL uptake after lipoprotein starvations. So the results show that the FIBP knockout, through the revolution of cholesterol biosynthesis and transport, rendered the CD8 positive T cells resistant to the immunosuppressive effect of high cholesterol, and this is a common feature of the tumor microenvironment. And finally, an anti-cholesterol drug named statins was introduced to the T cells. As you can see, the statin reduced the cholesterol level in T cells only before the activations, as shown in the figure 10A. However, after activations, statin treatment elevated the cholesterol level in T cells, as shown in figure 10B. Also, the statin treatment inhibited T cell proliferations and extended figure 10C and D, and consistent with a previous study that proved that statin inhibit T cell activation and proliferations. To a nutshell, the results suggest that a opposite effect between statin treatment and FIBP knockout in effector T cells was observed. While statin treatment elevated cholesterol level and reduced T cell proliferations, the FIBP knockout just inhibit the cholesterol metabolism and promote anti-tumor T cell responses. In the conclusion part, firstly, I would like to summarize the research flow of this study. First, based on the cytosine database, the TRACE platform evaluates the cytokine sensitivity by T cells in tumor environment. For example, the TGF-beta1 and PGE2 are common immunosuppressive factors, while TRACE is a triggering factor for the T cells' apoptosis mechanisms. And if the downstream pathways of the cytokine are activated, It means that T cells are under a very unfavorable environment. At the same time, the state health of T cells can be measured by expressions of cell cycle and DNA replication pathway genes. The repressed cells tend to have low activity in this pathway, and based on the interaction tests, the test model look for the T cells that remains healthy under pressure from various immunosuppressive cytokines. So the T cells are defined as tumor-resilient T cells, and the TRES for short. So these TRES signatures demonstrate important clinical applications. And based on the simplest coefficient calculations of systematically collected T cell genes expression profiling data, if the T cell sample are positively correlated with the resilient T cell profile, then the corresponding immunotherapy will achieve a good effect. In case there are negative correlations, the corresponding immunotherapy effect will be kind of poor. So in particular, the TRUST platform is almost completely accurate in predicting patients with poor outcomes in cell therapy using only the pre-manufactured samples. And the authors also analyzed the genetic characteristics of a TRUST using the single-cell expression data. The highest expression of FIBP genes in T cells almost overrated the low toughness of T cells, and in conclusion, the TRUST platform utilizes big data modeling to provide an important R&D tool for cancer immunotherapies, especially T cell therapies, and it was an important basis for guiding the clinical decision-making of the cell therapy later on. Finally, I would like to discuss about the achievements and limitations of this study. So the greatest achievement of this study was a TRES model that are able to provide proof of concept for identifying signature of tumor-resilient T cells. So no matter what you are, basic researcher, clinical researcher, clinicians, you are able to benefit from this model. And since the TRES model is able to well predict the cell therapy response, they may help patients to avoid unnecessary treatment that may entail in high cost and risky of side effects. The study also discovered FIPP genes as a novel negative marker of tumor-resistant cells in solid tumors. And the mechanistic study showed that the FIBP knockout enhances tumor and anti-tumor efficacy through down regulation, but not complete inhibition of cholesterol metabolisms, and indicate promise of combining immunotherapy with anti-cholesterol therapies. Since previous literature reported that the crowd inhibitory effect of FIB knocked out are observed in cancer cells, so the FIPP inhibitors may exert the anti-tumor effect in both cancer cells and T cells. Regarding the limitations of the study for the TRES model, the authors reported that when training test score, there are 38 out of 168 tumors did not achieve statistical significance. And the reason can be insufficient data quality, absence of tumor-resilient T cells in a tumor, or alternative immunosuppressive signal that not modeled by TRES. The interaction test that utilized by TRES assume that regression variables such as immunosuppressions and T cell proliferations should have suspicious variations. Therefore, the TRES model will not give meaningful results in highly immunosuppressive tumor, where all T cells are deeply dysfunctional without any variances, leading the covariate matrix in question closer singular. And for the FIPP genes, since poor knowledge about FIPP protein structure and biological roles confirmed, the other are unable to find any DNA or protein binding domain in the FIPP protein sequence. And they also suggest that more study should be done to evaluate the viability of FIPP as a therapeutic target. So thank you for your attention, and please feel free to ask any question regarding my presentation that you may have. Thank you.